All right, in this video, we'll be looking at membrane transport 2.6, and we'll just begin by talking about this concept of a concentration gradient. Concentration gradient is when there's more solute in one area than in another. And this class will mostly be dealing with a membrane in between those two areas. And so on this side of the membrane, you have a high concentration of solute. Remember, the solute is the thing that is being dissolved. And so in this case, blue hexagons are being dissolved in whatever the solvent is here, which is the thing that is doing the dissolving. Usually that's going to be water, and when we're dealing with cells. And so this here is a gradient: high concentration, low concentration. A membrane here is separating those two concentrations. You can have gradients in a lot of different setups, and concentration gradients are usually how cells are doing work, typically how cells will do work. That brings us to this concept of passive transport. Passive transport is molecular transport without using energy. The simplest kind of passive transport is called diffusion, in which you have a concentration gradient, area of high concentration, area of low concentration, and those materials will move from an area of high concentration to an area of low concentration. That is simple diffusion, requires no energy. These materials naturally want to move down here because they naturally want to achieve equilibrium, which is when both sides are equal, or you could even say when there's materials moving back and forth at an equal rate is another way of thinking about that. Another type of passive transport is called facilitated diffusion. It's diffusion moving from high to low concentration, except for this. At this point, there requires the use of a channel protein or a carrier protein we talked about in the last video. These materials are ionic or have some sort of polarity, and so they require help to get across the membrane, and so their passage across the membrane needs to be facilitated by a protein. And then there is active transport. Active transport is moving materials against the concentration gradient. So going from an area of low concentration to high. So pushing these orange diamonds across into a higher concentration requires energy. It's kind of like when you're asked to clean up your room and you decide that your entire room needs to go into your closet. Your closet can't fit all those things, yet you try hard and you push all the contents of your room into your closet. That requires energy. And that door is working really hard to keep everything in there. And you've also set up a concentration gradient, which there's a high concentration in your closet, low in your room. If you open that door, those things will naturally flow out into your room until all things are at equilibrium. You could harness that energy. You could do something with it. And that's what the cell will oftentimes do with concentration gradients, is use that energy that's built up to do some sort of cellular work. Here's another picture of active transport, just understanding if I want to move this plus sign into here, it's going to require energy because here is already full. And the movement of large materials, this is going to be called endocytosis. This is still a type of active transport in that it requires energy. However, in this particular instance, you have some large particle. In this case, you have like some sort of bean or something. This is maybe a bacteria. It's probably a more scientific thing. So here's the bacteria. This cell here, probably some sort of phagocyte or something, is going to come in and eat that bacteria. And you can notice what happens. It actually bends the plasma membrane around that bacteria and forms a temporary vacuole or vesicle in order to house that bacteria so that it can later fuse with the lysosome and eat it. And so endocytosis is the taking in of large materials. Particularities inside endocytosis: when it's solid particles, it's called phagocytosis. When it's liquid particles, it's called pinocytosis. And over here, you have receptor-mediated endocytosis, where there are receptors involved, and these receptors only bind to certain sorts of materials like stars, and it will take those stars in and do still endocytosis, breaks off part of the plasma membrane, forms a little vesicle in that, but it's looking for specific kinds of materials. And the opposite of this process is called exocytosis, when you have some sort of material that is made inside the cell. Take, for instance, maybe a protein. Cells have created proteins that those proteins need to go out to the rest of the body. Well, this little vesicle will be formed, and that vesicle will leave its contents will be pushed out into the extracellular space as the membrane of the vesicle fuses with the outer plasma membrane of the cell and pushes those materials out into the extracellular space. Again, this is a process that requires energy. This is not passive, but active transport.